start with the very first one, that is your gene mutation. Okay, so what happens is we know that genetic information is confined in your DNA, right? Okay, now in DNA, what happens is we have learned that in the DNA, the nucleotides are arranged in a specific sequence, right? Let's say A, T, C, C, G, C, A. Okay, clear?
Yeah.
So such kind of sequence is there in the DNA. Okay, now and all these sequences, they are responsible for a specific amino acid. We have learned that codons, like triplets, they code for one type of amino acid. Okay, clear?
Yeah.
And the same information is passed on when they make mRNA, and then using the information on the mRNA, amino acids are joined together. Okay. Now, if somehow if we change the information on the DNA, okay, now there could be multiple ways with which the information can be changed. Let's say if we delete this C, okay, this C has been deleted. Okay, clear? Or if we add some extra A or C over here, clear? Or let's say there is some rearrangement. This A came in the place of C and the C came in the place of A. Okay, so substitution of the position can also be can take place, right? Now all these things are actually taking place at which level? At the level of genes, right? Okay,
Yeah.
that's why it is known as gene mutation. But there can be other types of mutation as well, which are chromosomal mutation. Now, chromosomal mutation means a change in the sequence, basically the sequence of gene within the chromosome, or change in the total number of chromosome as well. Okay, what is the total number of chromosomes in human cell?
Is it no, it's 23 and 23, right? 46?
23 pairs. Okay,
Yeah, 23
how
pairs.
many
Okay.
we have? 23 pairs, right? Very good. So there are certain situations or cases where this number can go up and down. Means in some individuals they are born with 45 chromosomes. Some people are born with 47 chromosomes, right? So when these numbers also change, it can also cause mutations. Understood? Clear? The mutations can occur at different levels, at the level of gene, at the level of chromosomes, at the level of number of chromosomes as well. Okay, clear? So first of all, let's discuss that what do we mean by mutation itself. So a gene mutation is a change in the sequence of nitrogen bases in a DNA molecule. So what is the result of mutation over here? Mutations may result. See, again, there is the same thing that it may result in an altered polypeptide. May means there is a possibility, right? But many times it might not happen. Let's say, for example, we have a sequence. Okay, ATT, CG, G, let's say, whatever sequence is there, this is the sequence on DNA. During DNA copying also, let's say what happened, some error occurred, right? And let's say instead of this T, okay, C came. Clear? What came? C came. Okay, clear? There is still a possibility that this may still code for the same amino acid. Do you remember that we learned that?
Yeah, the code is degenerate,
Yeah,
right?
genetic codes are degenerate, means one amino acid can be coded by multiple codons, right? So let's say we had codon 1, which was coding for a protein X, okay, or amino acid X. After mutation, we got we got a codon that is 2, let's say 2, but still that is coding for the same amino acid X, right? So in this particular case, will there be any change in the polypeptide?
As long as it goes to the same amino acid, no.
No, right? Okay. So mutations may result in altered polypeptide. So it means there is mutation every time. Whenever a DNA replicates, there are mutations. Okay. And one of the most beautiful thing about our DNA is that 99% of our DNA is a waste. You know?
Waste?
Yes.
As in, like, it
Okay.
doesn't really code for
Clear?
anything.
So around just 1% of the DNA is your coding DNA.
Yeah.
Right? And around 99% is your junk DNA.
So what does the junk DNA even do?
Junk DNA keeps you safe.
Like?
Actually, like see, if you are coding this entire DNA, right? This entire means including the junk DNA. Okay. The chances of mutations are reduced. How? See, if let's say any mutation is happening here in any of these sites, right?
Yeah.
It is not going to affect us.
Yeah.
Understood?
Yeah.
It is not
Yeah.
going to affect us in any way. But imagine if you have the DNA and entire DNA, 100% of the DNA is coding DNA. Means error at any single point can cause problem to you. Okay. This junk DNA plays a very important role, right? Apart from that, we believe that the junk DNA or the non-coding DNA sequence, it has like informations, or you can say the secrets of your ancestors. Ancestors means long back when we used to be primates or apes, right? Okay.
Yeah.
So those genes which are not now non-functional, right? The information for those genes are stored over there. Okay. Anyway, so mutations can cause a different polypeptide to be synthesized or a protein to be synthesized. And because of the different polypeptide or protein, what will happen? You may have a different expression, right? Okay. Now, when this mutation can take place, it certainly can take place during DNA replication. Whenever cells are dividing, whenever your body cells are dividing, okay, and sometimes during DNA replication of your gamete formation. The gametes and gametes fuse together to form zygote. Okay, clear?
Yeah.
So there are two types of cells in the body. See, there are two types of cells in the body: somatic cells
And germatic
and
cells.
germ cells. Right? Okay, all the cells of your body which are non-reproductive. Okay, non-reproductive means they don't have any participation as such in reproduction. Okay, and they forms rest of your body. It forms the body of the organism. It forms what? Body of the organism. Okay, the skin, muscle, bone, blood, everything is your
Right.
somatic cells. Okay, clear? Germ cells are your reproductive cells. They are responsible for gamete formation. These cells divide to form what?
Gametes.
Gamete. Okay, clear? So when somatic cells multiply, they form your body. Okay, but when germ cells multiply, what they form? They form
Gametes.
gametes. Okay, somatic cells undergo mitotic division, while germ cells undergo, in some cases, mitosis is also there to increase their number, but in order to form gamete, they undergo meiotic division. Okay, clear? Anyways, the point over here is, let's say if any mutation has taken place in the somatic cell of your body, let's say, okay, a mutation occurred, let's see, when you are growing, your cells eventually multiply, right? Your muscle cells will multiply, okay, your skin cells will multiply, right?
Yeah.
Your blood cells will be synthesized. Okay, so in all these processes, let's say at any point of time, some of the cells got mutated. Okay, when any mutation takes place in the somatic cells, right?
Yeah.
That remains or goes away with that individual itself. Okay, these are non-inheritable changes. Okay,
So like cancer?
cancer anyway is not genetics. Okay,
Yeah.
you
Cancer
may
is
be
genetic.
sensitive. You may be sensitive to cancer. Means your body cells might be very sensitive to any specific kind of chemical. Okay,
Right.
let's say there is a family history. In that family history, we see that many of the members of the family, they suffered from cancer. So that is a clear indication that in their genes, okay, there are genes which are pretty much sensitive to some mutagens. Okay, clear?
Yeah.
It doesn't mean that if you have cancer, so your son or daughter will also have cancer.
Okay.
Okay, clear?
Yeah.
So we all have genes which can cause cancer in us, right? Those are called oncogenes. Which genes?
Oncogenes.
Oncogenes. Okay, oncogenic basically means cancerous. So we have oncogenes, and in some people they are more sensitive to certain mutagens. In some people they are less sensitive, right? So even if you are not exposed to any such mutagen, let's say you might have seen people who don't smoke, right? Or they don't even go out very oftenly, still they get certain kind of cancer, right?
Yeah.
Okay.
Yeah.
The reason being is their genes were actually sensitive to those mutagens. Okay. While there are some chain smokers, they keep on smoking throughout the day, but they don't get cancer.
Yeah.
Reason? Their genes are not that sensitive to that particular mutagen. Okay. So now let's talk about the different types of mutations. Right. So as already discussed, that some mutations only occur on the gene itself. Right. So let's say we have the DNA, and on the DNA we have certain sequences ATT, C, G, whatever. Okay. Clear. Now all these triplets codes for a amino acid. Right.
Acid.
Okay.
Hmm.
Fine. So because of any reason, let's just usually what happens is there are variations here as well. Okay. Let's say in some case that one of the nitrogen base is replaced by something else. Okay. Let's say the A over here is replaced by G. Right.
Yes, sir.
Okay.
Hmm.
So in this case, which all amino acids will be changed or altered? Let's say if I number 1, 2, 3. So which amino acids will be altered?
Just two. If it's substitution, it's just two.
It is 2, right? Okay. So change will be only at which level? 2. Okay. Clear?
Hmm.
Now imagine another situation where we have a DNA. In that DNA we have, let's say, a similar kind of sequence. Okay. Now, but this time instead of substitution, okay, means instead of being replaced, there is addition or deletion. Addition or deletion. Okay. Let's say here. Okay. Let's say this C has been deleted. Okay. So
Hmm.
what will be the effect of the deletion of C over here? First of all, let's try to figure out the new sequence. The new sequence will be ATT,
ATT, GAT,
then GAT,
QA onwards.
A, then whatever was there onwards, right? So here we have two different types of mutation. One is basically they all are point mutation, but in some cases what happens is the only place where alteration or change is going to take place is at that particular codon only. But in some cases where deletion or insertion is there, then you see a phenomena called frame shift. Which phenomena?
Frame
Frame
shift.
shift.
Frame shift, right? See why we are saying so, because if you see the second case, what you have seen, because of the deletion of the nitrogen base C, what you see is that rest all the genetic codes have been shifted, right? Or changed. So within the chromosome, sometimes what happens is the position of genes gets changed. Let's say here we have a gene, let's say A. Okay. During DNA replication, because of certain errors, what happened was the position of gene A is now changed to some other place. Okay. Clear? So when the position
Yeah.
of the entire gene gets changed, right? Here, what was happening in gene mutation? There was change in one or two nitrogen bases, right? But in chromosomal mutation, entire gene is changing its position. Okay, clear? And then we have whole chromosome mutations where the chromosome numbers gets changed. Instead of 46, you may have 45, 44, 47, 48. Okay, not 46, rest any other possibility. Clear? So whole chromosomal mutations are also there. So these are the three different types of mutation that we will be discussing. Okay. Okay. Now let's discuss point mutation. So what is point mutation? When only one or two nucleotides in a gene sequence are changed, right? Okay. So it could be substitution, it could be insertion, it could be deletion as well, right? So point mutation can be because of the following: substitution, where one base substitutes for another, while another is deletion, where a base is completely lost from the DNA sequence or gene sequence. And then comes insertion. So in insertion, there will be an additional nitrogen base. Okay, clear? So in the case of deletion, okay, and insertion, which phenomena will be observed?
The frame shift.
The frame shift, right? Okay, clear? So let's make instead of DNA, okay, let's make a DNA only. Write down any sequence, ATT, AGC, okay. So in the case of substitution, what will happen? Any
One
of the
base.
nucleotide, one base will
So
be
one particular mutated, like the codon will be changed, and that protein
changed.
may be changed, but sometimes it might not. That might not even happen. But like the rest
Right.
of the code following that mutation, vastly, it just remains the same.
Right. Okay. So here we can see that what happened is T is replaced by or substituted by A. Right.
Yeah.
Okay. Clear. A similar kind of thing we can do here as well in the next case, right? So in this DNA, let's say delete one of the nucleotide. Okay, so let's say we delete this T over here. Okay, here. So at
Yes, sir.
this particular point, we see that T, the codon T or the nucleotide T is deleted. Okay, and then lastly, we may insert something, right? Like ATT-GAGC. Okay, so here this G that came over here is your extra thing, right? Okay, so G is what? The extra codon. G is inserted. This is your extra codon, right? So these are some of the examples of the same thing. That now in all these cases, what will happen? The protein may get altered. Okay. Okay. So see, in substitution, we already learned, right, that what will happen, one nucleotide will be replaced with something else, right? So whenever such thing happens, usually what happens is that the change of amino acid is for that specific codon only, right? So a substitution mutation will only change the amino acid for the triplet in which the mutation has occurred, right? There will not be any change in rest of the amino acids. If you look at this particular diagram, so we have a genetic code and the amino acid sequence is something like this: tyrosine, serine, and leucine. Okay. Now, because of the substitution, this T has been substituted with A, right? Okay. And that change caused the substitution of serine with arginine. Okay, clear? But if you look at rest of the amino acid, they are still the same, right? Okay, clear? Now, in upcoming like slides, you will be discussed we will discuss about a disease called sickle cell anemia, right? So sickle cell anemia is also a very good example of same thing, right? Means point mutation. So in that also what happens is amino acid called valine, right, is substituted by another amino acid. Okay.
And done, sir.
Cool. What about you, Shreyas?
2 minutes. I'm at the stop codon, please.
Bro, I finished faster than you this time. Let's go.
Yeah, my Edexcel ran out of charge mid writing. Had to plug it in for like 2 seconds.
Honestly, that just sounds like a skill issue to me. So
Okay.
like,
Sure. 1 second, almost done. Okay, yeah, I'm done.
Okay, now insertion of nucleotides. So the same thing. A nucleotide will be randomly inserted anywhere within the DNA sequence. Okay, now because of this, what will happen? It will change the entire chain of amino acids, right? Okay, so these kind of mutation changes the amino acid that would have been coded for by the original base, as well as for rest of the other also a new base will be generated. Okay, clear? Let's look at this diagram over here. In the original sequence, we can see that the amino acid sequence is tyrosine, serine, and leucine. Okay, but because of the insertion of the new nucleotide here, now we can see that from this particular codon onwards, rest all the amino acid codons or amino acid sequence will be changed. So first is tyrosine. Now instead of serine, we have arginine. Instead of leucine, we have serine, right? And so on will be taking place. Okay.